Have you ever fried burgers in your lab? We have done that. And what that has to do with aroma analysis, we show you in this small video. Sizzling science with plant-based meat products. If we talk about measuring aromas from plant-based meat, we have to think about different analytical tasks. If you open the box, the burger patty should smell very well, even when you fry that in the pan. And of course, on the dish, the burger should smell also very good, and of course, it should taste good. The analysis is very challenging, so we have to deal with high concentrate water. We have to deal with sampling and sample preparation, and of course with low concentration of the aromas. Let us start with the high concentration of water. When we have a look to a burger patty, we have different ingredients next to the aromas. And the most of them are oil, texturous pea protein, and almost 60% of water. And does that affect our GCMS analysis? Normally, yes. But we have a solution for you. We use our Dynamic Headspace. And here you see a scheme of the Dynamic Headspace procedure, where we concentrate all the aromas and the water on the thermal desorption tube, and afterwards run a normal thermal desorption GCMS. But in our Dynamic Headspace, we have a drying station where we dry or where we purge out the water and keep the analytes on the thermal desorption tube. When we talk about sampling and sample preparation, it's a matter of where to sample and when to sample. When you open the box and even when you fry the patty, it's a matter of where to sample and how much to sample. But there's also a solution, because we fry our burger patty direct on the GCMS system in our autosampler in the Dynamic Headspace, in a closed system, so you don't lose any aromas or other analytes. Here we show you 5 aromas out of the complete analysis results we get. From the raw patty, we identified 3 aromas. In comparison to the frying pan, we have more aromas, but in less concentrations. And when we compare that to the Dynamic Headspace, we have much more aromas with much more intensity. This leads us to the low concentration problem. Aromas are really in low concentration, and the question is, how can we identify aromas in a GCMS? And you can do that with the Olfactometric Detector, which is a convenient and simple aroma sniffing detector. And here you see our ODI2 software, where we combine the MS data with the olfactometric data. And if you would like to learn more about this software, because there is so much more in, watch out our other video about the ODI2 software. For example, here you can see the olfactometric information shown in yellow, overlaid with the MS data in red. And this leads us over to a small concentration aroma, which we can smell, but which is not shown in the MS. And there is a solution for getting also this information on the MSD. It's called sniff and trap. So we use the olfactometric detector, and instead of smelling that, we place a thermal desorption tube during the time when the peak, which we can smell but which is not shown in the MSD, in the detector, and remove that after the peak eludes completely. And when we do that several times, we have a big concentration on the thermal desorption tube, and this thermal desorption tube can be analyzed in a normal thermal desorption MSD, and then you get the information from the MSD. You can identify this aroma. This is a very simple handling without having a very complex column switching device. And all these things makes your complex aroma analysis simple and relaxed with our Dynamic Headspace and the olfactometric detector. Watch out also our other videos and remain curious.